So later, then Hershey and Chase perform another experiment after the Griffith experiment in order to prove that yes, DNA is basically the genetic material which carries all the information of the life and also is responsible for carrying the character from one generation to the next generation. Uh, Hershey and Chase performed his experiment by using the bacteriophage. We know that bacteriophage is a viral particle that attacks bacterial cell. That's why it is called as the bacteriophage. Its life cycle, if we talk about its lytic life cycle, where when the viral particle attack on the cell, it replicates itself, and after the replication it makes several viral particles of its own kind. Then it ruptures the bacterial cell and all the viral particles they come out from the cell, and then again all those viral particles are available to attack the other cells. So at that time it was not established that whether it is only the DNA particle which gets into the bacterial cell or it's only the protein that gets into the bacterial cell, or both together they get inside the bacterial cell. क्योंकि ये कंफर्म नहीं था कि आया सिर्फ डीएनए इंटर होता है, प्रोटीन इंटर होता है इनसाइड द सेल ऑफ द बैक्टीरिया, क्योंकि उनको थोड़ा सा डाउट था कि अगर बहुत सारे वायरल पार्टिकल्स एक ही तरह के बन रहे हैं तो ये क्वाइट पॉसिबल है कि प्रोटीन कोट भी शायद सेल के अंदर इंजेक्ट हो अलॉन्ग विद वायरल डीएनए पार्टिकल, और उसी को देखते हुए बाकी वायरल पार्टिकल्स बन जाएं. So for that purpose, they perform an experiment. In that experiment, they basically perform two parallel experiments. In the one experiment, they use the phage and label their protein codes with sulfur 35 isotope. क्योंकि इस प्रोटीन में सल्फर अवेलेबल है, डीएनए के अंदर सल्फर नहीं होता. On the other side, they use another viral particle, and in that viral particle they label their DNA with the phosphorus 32 and allow both of these bacteriophage particle to attack on the bacterial cell. So after the attack, in one case where they basically label the protein code, they notice that there is no radioactivity inside the cell. Cell के अंदर कोई radioactivity नहीं है. But at the end, different viral particles of the similar structure were obtained. On the other side, where the Hershey and Chase labeled the DNA particle, in that case they noticed the radioactivity inside the cell. And at the end of the lytic cycle, several viral particles of the same structure and kind were obtained. तो दोनों में चूंकि वायरल पार्टिकल बन रहे थे और दोनों में सिमिलर वायरल पार्टिकल थे, तो एक चीज़ तो कंफर्म हो गई कि प्रोटीन कोट इंजेक्ट नहीं होता, सिर्फ वायरल पार्टिकल्स इंजेक्ट होते हैं, जो S35 से पता चला. और दोनों में प्रोटीन कोट सिमिलर स्ट्रक्चर के प्रोटीन कोट सिमिलर स्ट्रक्चर के वायरल पार्टिकल बनने ने ये इस्टेब्लिश कर दिया कि ये वाकई डीएनए है, पर्टिकुलरली इन दिस फर्स्ट इन दिस सेकेंड एक्सपेरिमेंट पैनल बी में, जहाँ पे ये कंफर्म हो गया कि सिर्फ वायरल डीएनए पार्टिकल के इंजेक्ट होने के बावजूद हेक्सागोनल स्ट्रक्चर के बैक्टीरियोफेजेस बन रहे हैं. तो इट मीन्स दैट दिस डीएनए बेसिकली contains all those information which are required to make the complete set of a viral particle. So after establishing, then later several experiments were performed to know that what is DNA basically. So DNA is basically a polynucleotide chain. Polynucleotide chain होती है DNA, or in which a single unit is called as the nucleotide. Our nucleotide is composed of nitrogenous base, pentose sugar, and phosphate group. So if only this sugar and nitrogenous base is linked together, so this is called as the nucleoside. And when this phosphate group it joins this nucleoside, it make a complete unit structure which is called as a nucleotide. If we talk about the nitrogenous bases, there are two categories of the nitrogenous bases: purine and pyrimidine. Purines are a double ring structure. You can see here in this structure that one of the 6 corner ring and the other of the 5 corner ring. and pyrimidine is a single ring structure, you can see here. Purines are of the two types, adenine and guanine, and pyrimidines are of three types, cytosine, thymine, and uracil. Cytosine and thymine is the component of the DNA, and uracil is the component of the RNA. Nucleotides again are of the two types, deoxyribonucleic acid or deoxyribonucleotide. So deoxy means that nucleotide where the pentose sugar is deficient of a single oxygen at position number 2.